Hi, my name is Dave Brown. I'm a retired veterinary virologist and my PhD is in genetics. During my career, I have worked primarily in the area of infectious disease, studying genetic influences on the ability to resist virus-induced cancers, using recombinant DNA techniques to develop vaccines and assays for virus detection, and teaching virology to veterinary students and molecular biology to graduate students. I'm telling you this not to toot my own horn, but because I think you should be skeptical about your source of information, and that's exactly what I am attempting to be, a source of information. I'm not an MD, and I would want you to definitely consult your healthcare professionals before making changes in medication or taking large doses of supplements that could potentially impact the efficacy of your medications. I am merely trying to communicate what I feel is critical information in this time of crisis, information well known to many in the scientific community, but that has unfortunately not filtered down to the general public. This cartoon by Gary Larson perhaps explains it better. There is critical information available that is obvious to many scientists, like an elephant in a room. I'm merely trying to help you find that elephant. This information comes from the website of the Centers for Disease Control and Prevention. It contains recommendations that we have all heard over and over again. The respiratory hygiene refers to the wearing of masks. Good advice. But there is a very important prevention tip that is missing, and most of the rest of my talk will address that critical bit of missing information. Here is that elephant, out in plain sight, and here is its simple message. Get your vitamin D levels up, and do it before you reduce your social distancing or lower your vigilance in following the CDC's recommendations. The strategy to increase individual resistance to COVID-19 by improving vitamin D status is not a substitute for a careful, well-planned reopening of the U.S. economy, as described by Dr. Fauci. Vitamin D should be considered an additional preventive measure to strengthen resistance to multiple pathogens, including COVID-19. Much of my talk will deal with this hypothesis. Seasonal fluctuations in vitamin D levels are a major driving force behind the seasonal expression of viral pathogens, including human coronaviruses that cause the common cold, and for winter outbreaks of influenza. I will show you evidence that the same goes for COVID-19. The situation with the current outbreak of COVID-19 may improve as the sunny days of summer return, but not for the reasons suggested by our president. Many of us have been cooped up indoors during this critical time. We can make our own vitamin D, the sunshine vitamin, but it depends on exposure to particular wavelengths of ultraviolet light from the sun. When the sun is low in the sky, ultraviolet light is filtered out by the atmosphere. It's also filtered out when light passes through your window panes. So you won't get your much needed vitamin D by sitting next to your window, at least if the window's closed. But we can rapidly and safely improve our vitamin D status through supplementation. However, you won't likely obtain enough using a daily multiple vitamin pill. Most of us in the North do not have the luxury of going south every week and spending hours in the sun, or of having weekly sessions in the tanning booth. As little as our president has done to prepare our nation for this COVID-19 pandemic, he has actually spent many hours preparing himself. This slide shows the increase in the number of symptomatic COVID-19 infections in this country, starting with a few cases at the end of January. 3 months later, we have over 1 million cases. The column on the right shows the number of new cases added each week. The good news is that the number of new cases added each week appears to appears to have stabilized. The bad news is that we are still dealing with a huge number of new cases. Unless adequate precautions are taken, that number could blossom exponentially, just as it did in March. This is just a snapshot of the COVID-19 pandemic taken on April 24th. I've highlighted a couple things. The U.S. has not fared well, and the caseload and death toll continue to climb. But look at Australia and New Zealand. They have had many fewer symptomatic cases, and the fatality rate associated with those cases is much less. I'm sure that their rapid response to contain the infection helped, but that would not explain such a lower death rate. Is their health care system so much better at treating these infections? I doubt it. Most likely vitamin D status has played a major role. At the time of the outbreak, their citizens would have benefited from months of summer sun. In a couple months, New Zealand and Australia will be experiencing their winter. If vitamin D levels are not maintained through supplementation, they may suffer a more deadly outbreak if exposed to another round of the virus. This slide shows the number of COVID-19 cases as of May 4th. For both the Northern Hemisphere and Southern Hemisphere, I've listed 8 countries with the greatest number of cases. You don't need to do much of a statistical analysis to see a tremendous difference between how much worse we in the Northern Hemisphere have fared compared to our Southern brethren. At the outset of this pandemic, folks in the Southern Hemisphere had just enjoyed months under the summer sun, while we had bundled up bearing the cold or snuggled up indoors during the long winter nights. This slide focuses on the mortality rates we have experienced compared to the outcomes in Australia and New Zealand. The differences are dramatic. We have a staggeringly high caseload compared to Australia and New Zealand, and adjusting for population size, the number of cases per million people in the U.S. is still tenfold greater than that reported in Australia or New Zealand. The death rate in the U.S. at 5.7% is 4 to 5 times as high as in Australia and New Zealand. As I am sure you are aware, the elderly are more susceptible to severe outcomes of COVID-19 infections. So I have provided a statistic showing the percentage of each country's population that is 65 years of age or older. The percentage of senior citizens in Australia and New Zealand is just as high or even higher than in this country. So why is the death rate so much lower than here in the U.S.? Is the Australian healthcare system so much better, or are the citizens in Australia and New Zealand that much healthier? Perhaps. But due to the time of year, the vitamin D status in the U.S., especially northern parts of the U.S., would be lower than in countries of the southern hemisphere. This graph from the CDC documents the more severe outcome of COVID-19 infection associated with advancing age. There are certain health conditions that are associated with increased risk of severe COVID-19 infection. Many of these conditions are treatable by lifestyle changes, but the U.S. approach to medical care relies heavily on the pharmaceutical management of symptoms rather than cure. There are a number of drugs that can adversely impact vitamin D status, including statins. For drugs that are commonly prescribed in this country, it would be prudent to consider the pharmaceutical drug usage as a potential risk factor, not just the medical condition being treated. This is another graph from the CDC. If you look at the non-Hispanic black population, you will see that although blacks represent only 18% of the population being studied, they represent 33% of the COVID-19 hospitalizations. They are clearly overrepresented. The poor and the minorities are so often underrepresented in our government. In this case, overrepresentation is clearly not a good thing. We need to solve this situation and fix it fast. The good news is that I think we can fix it fast and save many thousands of lives in the present and millions of lives in the future. One explanation for increased COVID-19 risk with the black population is that in many ways, severe COVID-19 infections represent another disease of poverty. Poverty certainly plays a role in creating conditions that harm health and potentially limit response to the virus. But I believe there is much more to it than that, and that many wealthy blacks with good diets and great health insurance will remain at high risk unless a critical risk factor is addressed. This slide compares a number of countries. Greatest number of cases per million people has occurred in the U.S. by far. A high GDP does not offer a better outcome. The population of Indonesia approaches that of the U.S. With a GDP 1/6 that of the U.S., the number of cases per thousand is 1% that of the U.S. It is highly unlikely that the more favorable outcome in the Indonesian population is the result of greater economic security or better health coverage. These are risk factors for severe COVID-19 infections, as reported by the CDC. And some conditions like obesity and diabetes are found with greater frequency in poor populations in the U.S. Many of these conditions are quite common in our country, which could, at least in part, explain the greater risk of infection and death that we have experienced. There is great concern for the high incidence of severe COVID-19 infections occurring in the black population. This table lists some logical explanations offered in the media, but I have added an additional risk factor that I believe is most important in terms of outcome of COVID-19 infection. Greater pigmentation of the skin protects from the damage caused by ultraviolet light, but it also reduces the amount of vitamin D produced by a given level of light. This chart shows that people with the darkest skin come from Zone 1, where there is greatest level of ultraviolet radiation. Paler skin evolved in areas with lower levels of sunlight to improve vitamin D status. For those of us who bemoan our pale condition, we should probably appreciate the improved vitamin D status it may promote. In the UK, we also see an overrepresentation of people from minority ethnic backgrounds among those with severe COVID-19 infections. The BBC reported that 34 of 4,800 critically ill patients came from minority ethnic backgrounds, whereas they represented only 14% of the population. Indians in England were among the worst affected, and it was suggested that more crowded living conditions may have played a role. But among Indians living in India, where living conditions can be quite crowded, the COVID-19 death rate is considerably lower than in the US or UK. Unlike the US or UK, a high percentage of cases were among younger people, less than 40 years old. Yes, India is a younger crowd, and this could have had an impact. This table shows the age distribution by country for India, the UK, the US, and Australia. The age distributions in the US, UK, and Australia are very similar. Only 6.2% of the population in India are seniors. But India has a large population, and that translates to a large number of potentially vulnerable seniors. There are far more seniors in India than in either the UK or US. This slide shows that despite the greater number of seniors than in the US and UK combined, the number of fatal COVID-19 infections is minuscule compared to those occurring in the UK or here in the US. Why? Why do seniors in the US and UK appear to be less resistant to severe COVID-19 infections? I've listed a few possible explanations. Perhaps our seniors have a greater number of health-related risk factors. Perhaps the frighteningly high numbers of prescription drugs our seniors take are having adverse effects. Perhaps India did a better job of controlling viral spread by responding in a more timely fashion. But coming as no surprise to you, I believe that our low vitamin D status here in the U.S. is a major factor responsible for our much poorer outcome in this pandemic. This preprint of a paper from the Philippines became available online in April. Look at the title. The idea of using vitamin D supplementation to increase resistance to the COVID-19 virus is not just my idea. Many scientists believe this. In China, clinical trials are in progress to evaluate vitamin D as a therapy for COVID-19 associated disease. High doses of vitamin C are also being considered. It is surprising that the potential value of both vitamin D and vitamin C is rarely, if ever, discussed by mainstream media. But we hear a heck of a lot about injections of disinfectants and the use of pharmaceutical drugs with negligible to modest efficacy. In this chart, you see this table labeled as mean serum 25-OH-D. That represents the average amount of 25-hydroxyl vitamin D in the blood. And this form of vitamin D is the major and most stable form of vitamin D. Vitamin D has to be modified by the addition of a couple hydroxyl groups in order to become active. I'll talk about that in a little bit. But as you can see here, in cases where the outcome of infection was mild, the average concentration of vitamin D was 31.2 nanograms per mil. Notice that the lower the level of vitamin D in the serum, the more severe the outcome. There is a controversy concerning what is considered an adequate level of vitamin D. I'll get to that later, but the Institute of Medicine claims that 20 nanograms per mil is sufficient. Perhaps in regard to bone formation, higher levels of vitamin D are not required for some people. But in this study, just a 10 nanogram per mil increase was associated with a mild versus severe outcome of COVID-19 disease. Repeated tests for vitamin D levels were taken on these patients, and lower vitamin D status was present at the onset, not a consequence of the outcome. Looking just at vitamin D status, it was reported that patients having a vitamin D level of less than 20 nanograms per mil were nearly 24-fold more likely to have a critical rather than mild outcome. That is remarkable, and it suggests an impact on disease severity far greater than offered by any drug currently being considered. I doubt that even vaccination will provide such protection from severe disease, at least for the most vulnerable of our population. A recent preprint of a paper from Indonesia had similar findings. This study involved 780 cases of laboratory-confirmed COVID-19 infections. The majority of deaths were male, older, had lower than normal vitamin D levels, and they had pre-existing conditions. Without adjusting for other risk factors, the risk of death was 19 times higher for cases with a deficient vitamin D status, less than 20 nanograms per mil, as compared with a normal vitamin D level of greater than 30 nanograms per mil. This table shows the relative risk after adjusting for age, sex, and comorbidity. The risk of death is still 10 times higher for a deficient vitamin D status and nearly 8 times higher for an insufficient status compared to those with a vitamin D level greater than 30 nanograms per mil. This chart shows the average vitamin D level associated with U.S. residents in the northern and central states versus southern states in the winter and summer. Levels are up in the summer, but not spectacularly high. In winter months in the northern U.S., the mean vitamin D level of 21 nanograms per mil is almost exactly that associated with a severe outcome of COVID-19 in the Indonesian study. There are seasonal patterns associated with a variety of pathogens. Some viruses, like West Nile virus, are transmitted by biting insects. Diseases caused by these viruses are seen at times of the year when the insects are around. Some viruses announce their appearance during the winter months. Acute respiratory illnesses in general are prevalent during the winter months, at least in temperate climates, and influenza virus is a classic example. Many of you may also be familiar with norovirus, also known as winter vomiting bug. It has a reputation for occasionally lessening the lure of the luncheon buffet of cruise ships. A highly infectious non-envelope virus, its seasonal pattern is very much like the envelope influenza viruses and coronaviruses. It can persist for weeks in the environment and resist temperatures of 140 degrees Fahrenheit. This slide and the couple that follow come from a classic review paper by John Cannell and colleagues. The table shows the weekly consultation rates for illnesses diagnosed as influenza or influenza-like. Based on data collected from about 40 general practices in the UK from 1968 to 1970, it shows the classic pattern of low frequency of illness in the summer months and peaks of infection in the winter. This table shows seasonal variation of 25-hydroxy-D levels in inhabitants of a small southern German town. Lowest levels of vitamin D are seen in January, February, and March. Even as late as June, levels of vitamin D are below 20 nanograms per mil. Peak levels of vitamin D occurred in August and September, lagging from the time of peak solar radiance in the latter part of June. This diagram shows by month the percentage of each zone's type A influenza outbreaks. In the temperate zones, the majority of outbreaks occurred during the winter months, December, January, February, March in the northern hemisphere, and June, July, August, September for the southern hemisphere. This very interesting paper from South Korea reports on the prevalence of norovirus in children in both winter and summer. This highly infectious and environmentally stable virus was found in 3.5% of asymptomatic children tested in the summer. So why aren't we seeing clinical disease in these kids? And why is there such a distinct seasonal pattern for this virus? By now, I think most of you have heard of the famous 1918 to 1919 influenza pandemic that killed so many people around the world. This paper presented evidence that the fatality rate in northern cities of the U.S. was higher than further south. For example, the fatality rate in New London, Connecticut was over 3 times that in San Antonio, Texas, and the frequency of pneumonia complications was double. The authors hypothesized that lower levels of vitamin D in inhabitants of northern cities may have played a role. Returning to the 21st century, and another study involving inhabitants of Connecticut, this paper evaluated vitamin D status of healthy participants at various times during the fall and into January. The incidence of acute viral respiratory tract infections and the average number of days sick were lower for participants who maintained a vitamin D level of 38 nanograms per mil or higher throughout the test period. Only 3 of 18 participants with higher vitamin D levels became ill, as compared to 81 of 180 participants with lower levels of vitamin D. Everyone agrees that vitamin D is essential, but different groups of scientists and medical practitioners have different opinions concerning what is optimal. The Institute of Medicine considers a value greater than 20 nanograms per mil as sufficient. The Endocrine Society considers a value greater than 30 nanograms per mil as sufficient. The Vitamin D Council considers a value of over 50 nanograms per mil as sufficient and 50 to 80 nanograms per mil as optimal. I personally would go with the Vitamin D Council's recommendation. In the previous slide, we saw that participants with vitamin D levels of 38 nanograms per mil or higher had a significantly lower incidence of viral respiratory tract infections and shorter durations of illness. This graph comes from data from the National Health Nutrition Examination Survey. Vitamin D levels were low for the vast majority of participants, but especially low for black subjects. Keep in mind that 30 nanograms per mil and higher was associated with greater resistance to acute viral respiratory tract infections in the earlier slide. By that standard, only a small minority of the participants would have had vitamin D levels in the protective range. Even by the conservative standard of the Institute of Medicine, the vast majority of blacks surveyed would be considered deficient in vitamin D. Our healthcare system has done very little to improve this serious situation, and many people, especially non-white minorities, have suffered poor health as a result. Because blacks have a lower risk of fragility fractures than whites, it has been argued that they don't need higher levels of vitamin D. But the minimum level of vitamin needed to spare bones is likely not the same as for other important functions of this vitamin. In another study, it was reported that blacks with 25-hydroxyl D levels greater than or equal to 20 nanograms per mil were at higher risk of developing fractures, and concern was expressed that blacks might be more susceptible to vitamin D toxicity. In this slide are results of two studies conducted in Africa. The upper study looked at vitamin D levels in 123 pregnant women from Gambia. The mean vitamin D level was 44 nanograms per mil, with a range of 35 to 76 nanograms per mil. These women were not suffering fractures. In the bottom study, looking at people living traditional lifestyles, the mean concentration of vitamin D for the Maasai was about 48 nanograms per mil, and for the Hadzabe, 44 nanograms per mil. I doubt that any of these people were suffering fractures or any disease manifestation of vitamin D toxicity. It is absurd that there is so much concern about vitamin D toxicity in this country when the evidence points to severe consequences of widespread vitamin D deficiency. This is a clinical trial involving 400 infants randomized into low-dose and high-dose vitamin D groups. A significantly lower incidence of influenza infection and a shorter duration of symptoms was associated with the high-dose group that received 1,200 units of vitamin D daily. The American Academy of Pediatrics recommends 400 units daily for infants. This is data from the CDC concerning the 2017 to 2018 influenza season in the U.S. Among seniors, there were nearly 6 million symptomatic cases, with an estimated death rate of nearly 1%. Among people aged 50 to 64, there were over 13 million cases, but a much lower death rate. That's 19 million cases in just the 50 and older age group, despite the widespread use of a vaccine for influenza. COVID-19 is associated with a considerably higher fatality rate, and due to its ability to be shed for up to 2 weeks before onset of clinical symptoms, rapid spread of the virus is expected. Should we be putting all our hope in a vaccine? This is the compilation of 10 seasons looking at effectiveness in the community-dwelling elderly. Influenza vaccination was associated with a 27% reduction in the risk of hospitalization for pneumonia or influenza. What about the other 73%? How can we do better? Of course, you know how I think we can do better, but how can a vitamin for building better bones help? Well, vitamin D does a lot more than help build strong bones and teeth. Here are a list of conditions associated with low vitamin D status. Almost every common disease plaguing our society is connected in some way with a vitamin D deficiency. I've highlighted immune system diseases and inflammatory conditions because of vitamin D's well-established roles in regulating the immune response and helping prevent damage from excessive inflammation. Uncontrolled inflammation underlies most of our chronic health conditions, so loss of proper regulatory control because of insufficient levels of vitamin D could certainly contribute to a wide variety of diseases. So I think in most of the disease listed, the association is causal. For kidney and liver disease, vitamin D deficiency can be a consequence, since activation of the vitamin to its active state occurs in these two organs. But how can a single vitamin be involved in so many different diseases? I think I need to give you some background about this vitamin to better answer that question. So what is vitamin D? These drawings depict the structure of two forms of vitamin D. One from plants, known as ergocalciferol, or vitamin D2. The other, called cholecalciferol, or vitamin D3, is a type made in animals, and it is the type that I recommend as a supplement. There are slight differences at the upper right end of the molecule. Both forms are used as supplements, and it is just my bias to prefer the type our bodies are used to using. There are also some recent studies to suggest it is the better choice. The UVB portion of light interacts with a precursor in fungi to form ergocalciferol, vitamin D2. In animals, the precursor located in the skin is 7-dehydrocholesterol, and the product produced following exposure to UVB light is cholecalciferol, vitamin D3. I'll refer to vitamin D3 as vitamin D for the rest of this talk. We call vitamin D a vitamin, but it is really a hormone precursor. Unlike your typical vitamin, it is not an essential component of our diet that we can't synthesize ourselves. As long as we have adequate exposure to the right wavelength of UV light, our bodies can manufacture our own vitamin D. But until vitamin D is activated by the additions of two hydroxyl groups, one OH group added to each end of the vitamin cannot perform its hormone-like activities. The first hydroxylation step occurs in the liver to form 25-hydroxy vitamin D. This is the most stable form of vitamin D and the one that is usually measured in blood tests. The second hydroxylation step occurs in the kidney to form the active hormone 1,25-dihydroxy vitamin D, also known as calcitriol. This second hydroxylation step can occur in other locations of the body, such as in cells of the immune system, and the localized concentrations of calcitriol that result can regulate the expression of a large number of genes in a wide variety of tissues. In an earlier slide, I showed an association between vitamin D deficiency and kidney and liver disease. In these cases, the vitamin D deficiency could potentially be a consequence of the diseased organ, since these organs play important roles in activating the vitamin. Kidney disease can cause a condition known as renal rickets. Apparently, cells actively involved in bone formation are dependent on vitamin D that has already been converted to calcitriol in the kidney. After activation to form calcitriol, this activated form of vitamin D enters cells and binds with a vitamin D receptor and another vitamin A-related receptor. This complex then enters the nucleus of the cell and binds with specific sequences on the chromosomes. This results in transcription. That's the formation of messenger RNA, which is subsequently translated into proteins in the cytoplasm of the cell. In some cells, such as certain cells of the immune system, the inactive 25-hydroxy-D can be converted to calcitriol, allowing it to bind to the vitamin D receptor. I realize this is getting a bit beyond many of your backgrounds. But the takeaway that I would like you to retain is that activated vitamin D is a hormone, and that it plays an important role in regulating gene expression in a wide variety of tissues. That is why a deficiency of this single molecule can adversely impact so many organ systems. The second takeaway is that in order for calcitriol to function, it must bind to a vitamin D receptor. It may be significant that there are variants of the vitamin D receptor, and people bearing some variants may require higher levels of vitamin D. There's evidence, which I won't present in this talk, that some of these people may be at greater risk of developing diseases, such as tuberculosis, leprosy, and multiple sclerosis. As I mentioned, regulation of many gene products is via the vitamin D VDR complex, but the story gets a bit more complicated. Some gene products need further modification for them to become active. I'll just briefly talk about two gene products that are particularly important for proper tissue calcification. Osteocalcin and matrix GLA protein are proteins expressed under the control of the vitamin D VDR complex. Both proteins must be carboxylated in order to become active. Vitamin K2, another important fat-soluble vitamin, acts as an enzyme to promote the carboxylation step. Osteocalcin promotes bone mineralization, a process that is sorely needed by many in our society. But one thing we don't need is calcification of our arteries. When matrix GLA protein is carboxylated by vitamin K, it helps prevent inappropriate calcification of soft tissues. I take quite a bit of vitamin D daily, and I also take a vitamin K2 supplement to reduce the danger of inappropriate calcification of soft tissues. Most immune cells, as well as cells lining blood vessels and the respiratory tract, have an enzyme that can transform 25-hydroxyvitamin D into calcitriol. This may be particularly important for a well-regulated and comprehensive defense against respiratory pathogens like COVID-19. I list a few relevant activities of vitamin D, but what I would like you to understand is that the magnitude of the immune response is not necessarily the best indicator of its efficacy. The immune response must be properly regulated. If not, the response can evolve into a severe and potentially lethal inflammatory state. This appears to be happening in some cases of COVID-19 infection. What is called a cytokine storm develops, causing considerable inflammatory damage and triggering the formation of blood clots. Vitamin D helps mobilize the initial innate immune response that helps control viral spread while our bodies prepare for an adaptive response. But it also helps prevent an excessively inflammatory state by inhibiting proliferation of certain lineages of T helper cells that secrete certain inflammatory cytokines. These are some mechanisms our bodies use to resist infections. Viruses need live host cells in order to replicate. Our skin is covered by a layer of dead cells. A virus can get by that barrier if it can be spread by a blood-sucking insect like the mosquito. The integrity of the mucosal barrier plays an early role in limiting viral access to target tissues. Our intestinal tract and respiratory tract are lined by a mucosal barrier. Inflammation can cause increased permeability of that barrier. Innate immunity involves recognition of molecules commonly associated with various microbes. An adaptive immune response involves the recognition of specific antigenic epitopes by a few cells, followed by an expansion of those cells capable of binding with the antigenic peptide. I'll try to give you a little immunology background to help you understand the multiple ways vitamin D can help promote resistance to infection. This slide shows vitamin D regulating both the innate immune response and the adaptive immune response. Certain cytokines are being suppressed, and a Th2 type of response is being promoted. This type of response stimulates the production of antibodies by B cells. A Th1 response is being blocked. Th1 cells promote a response dominated by cytotoxic T cells. These cells kill virus-infected cells. Perhaps this type of response may be more likely to produce excessive levels of inflammation, but that is just speculation on my part. Earlier days in my career, I would have thought that suppression of the cytotoxic T cell component of the immune system would be a bad thing. But when considering the thin linings where gas exchange in the lungs occurs, excessive inflammation and fluid accumulation within tissues may have more severe consequences, essentially causing death by suffocation. The immune system is very complex, and there is still a lot of important information being discovered. One should pause before thinking that the many complex activities regulated by vitamin D can be replaced by a few pharmaceutical drugs. If a deficiency of vitamin D is causing problems, it is ludicrous to try to solve those problems without trying to correct the vitamin D deficit. But that appears to be the approach our country has taken. In this slide, I am just showing some ways vitamin D could help protect against severe COVID-19 infections. It helps maintain the integrity of mucous membranes. Not only does that help reduce spread of the virus, but it helps retain the structural framework needed for efficient gas exchange. Vitamin D enhances innate immunity, slowing early progression of the viral infection. It stimulates the production of antimicrobial peptides that can be effective in reducing the risk of pneumonia caused by bacteria, and a secondary bacterial infection is a real concern. As I mentioned before, the complex regulation of the immune system, orchestrated by vitamin D, can promote a protective antiviral response in the lungs, and at the same time prevent excessive inflammation that could precipitate a cytokine storm. And perhaps of great significance for COVID-19 infections, it could reduce the risk of blood clot formation. Vitamin D has been found to decrease platelet aggregation. Clumping of these tiny platelets around a damaged blood vessel would help in the formation of blood clot, also known as a thrombus. That could help prevent hemorrhaging, which we don't want. But if the thrombus is large enough, it could block the blood vessel. This is what occurs in a stroke, and we certainly don't want that. Aspirin decreases platelet aggregation, and it reduces inflammation. Does that mean we would do just as well or better using aspirin or another drug to try to prevent this deadly consequence? And this consequence is a real concern, as it appears to be a major mechanism contributing to a fatal outcome of COVID-19 infection. I don't know the answer, and many doctors are struggling now with finding the right answer, since an excessive dosage of an anticoagulant can cause fatal widespread hemorrhage. Vitamin D also causes increased levels of a molecule called thrombomodulin. Thrombomodulin is a membrane protein expressed on the surface of endothelial cells that line blood vessels. It reduces blood coagulation by converting thrombin, a procoagulant enzyme, to an anticoagulant enzyme. Endothelial cells express the vitamin D receptor, and they have the enzyme needed to convert 25-hydroxyvitamin D to calcitriol. Vitamin D also decreases levels of tissue factor from immune antigen presenting cells called monocytes. Tissue factor from monocytes and macrophages causes thrombin generation. Excessive expression of tissue factor, which can occur in a poorly regulated immune response, can lead to a condition called disseminated intravascular coagulation. So vitamin D reduces risk of excessive blood clot formation through multiple mechanisms. It seems plausible that both increased susceptibility to infection and risk of severe complications of infection are due to a widespread deficiency of this essential vitamin. Using vitamin D to correct a deficiency makes sense to me, not only for prevention, but also for treatment. Many people in this country suffer from a condition called inflammatory bowel disease. With this condition, intestinal hyperpermeability, also referred to as leaky gut, leads to exposure of the immune system to intestinal contents normally blocked by a healthy epithelial barrier. This increased exposure to bacterial products and other foreign substances leads to a constant state of inflammation. In this study, the authors used vitamin D to treat experimentally induced colitis, inflammation of the colon, in mice. Vitamin D markedly increased expression of tight junction proteins, leading to decreased permeability of the intestinal epithelial barrier. These tight junctions also play an important role in maintaining pulmonary barrier integrity. The authors in this paper demonstrated that vitamin D receptor deficient mice have decreased levels of the tight junction proteins. Remember that it is the vitamin D VDR complex that regulates the expressions of genes involved in maintaining the epithelial barrier. It is not a direct effect. That is why a single molecule can have so many effects. Vitamin D is a team leader, directing many players on the field to reach that goal of optimal health. When we lose that leader, we also lose many players on the field. We can try to replace a few of the players using a drug approach, but it is not the approach that nature provided us, an approach that has worked pretty well for millions of years. Diabetes patients can suffer tremendous damage to the endothelial lining of their blood vessels. It can eventually compromise blood flow so much that it necessitates the amputation of limbs. This is not because of high blood cholesterol levels. It's because of the real culprit behind the high incidence of cardiovascular disease in this country, high blood sugar levels. Through a process called glycation, high blood sugar levels result in sugar binding to the surface of the endothelium and initiating an inflammatory response. Platelets aggregate at the sites of the inflammatory damage. This first paper just shows that platelet aggregation is increased in type 2 diabetes patients with an insufficient vitamin D status. Hemoglobin A1C is a measure of the amount of glycation that has occurred in red blood cells over their lifespan of several months. An inverse relationship with vitamin D levels suggests that vitamin D also helps prevent glycation, a step preceding platelet aggregation. The second paper on this slide reports a 31% incidence of thrombotic complications in intensive care unit patients with COVID-19 infections. This remarkably high incidence is undoubtedly contributing to the high incidence of fatality we are seeing with our COVID-19 patients. So a second underlying condition that is pervasive in our society is likely contributing to our poor outcome with this virus. Insulin resistance is rarely addressed until it is severe enough to elevate blood glucose levels enough to catch our doctor's attention. But damage to our blood vessel walls is occurring well before that. An elevated LDL cholesterol and decreased HDL cholesterol is a message that something bad is happening. It is not the cause. I am going to digress for just a minute. We saw earlier that vitamin D reduces risk of thrombotic complications in several ways. This study investigated levels of vitamin D in patients with an autoimmune disease that's characterized by the development of antibodies against phospholipids. These antibodies can increase the risk of blood clot formation. Frequently, this manifests as deep venous thrombosis in the legs. 115 individuals with the syndrome, known as the antiphospholipid syndrome, had an average summer vitamin D level of 28 nanograms per mil, compared to 40.1 nanograms per mil in healthy controls. Thrombotic patients had a mean vitamin D level of 20.8 nanograms per mil. Low-dose vitamin D therapy in a few patients did not appear to be an effective treatment. Many people are at risk of developing blood clots, and they are often on medications to thin the blood. Warfarin, an anticoagulant used in rat poison, is a common medication used to slow clotting time and decrease the chance of thrombus formation. It works by inhibiting carboxylation by vitamin K and the activation of some clotting factors. So people on warfarin are told to avoid green leafy vegetables that are a rich source of vitamin K1. But various forms of vitamin K2 can also potentially carboxylate and activate clotting proteins. Long-term supplementation with higher doses of vitamin D3 should be accompanied by vitamin K2. So people with blood clotting abnormalities, a logical strategy would be to adjust their medication amount or the type of medication to allow daily and consistent supplementation with the critical health promoting combination of vitamins D3 and K2. We need to do what's best for the patient, not what's best for the medication. Cholesterol has been given a bad rap. It is essential for life, found in all cells of the body. It is a precursor to hormones, vitamin D, coenzyme Q10, and a building block for cell membranes. It is needed to repair cell membranes, and low-density lipoprotein transports cholesterol from the liver to other tissues of the body, while HDL transports cholesterol back to the liver. A high ratio of LDL to HDL cholesterol likely reflects an ongoing need for tissue repairs. In the case of cardiovascular disease, tissues needing repair would be the endothelial lining of blood vessels. So this high ratio of LDL to HDL most likely just reflects an ongoing process, such as glycation of endothelial surfaces that could compromise future health. Our medical system is heavily influenced by the pharmaceutical industry. There's a pill for just about everything. Statins are prescribed to lower cholesterol levels with, at least in my opinion, the misguided belief that the culprit behind cardiovascular disease is cholesterol. By inhibiting cholesterol synthesis, some levels of other important molecules that are derived from cholesterol, like vitamin D, coenzyme Q10, estrogen, and testosterone, can be compromised. A number of people have adverse reactions to statin therapy, so other drugs are being evaluated to lower LDL cholesterol and raise HDL cholesterol. I believe this is a case of shooting the messenger. Among the new drugs being evaluated are cholesterol ester transfer protein inhibitors. Great results were anticipated for CETP inhibitors. They do a better job of improving the total to HDL cholesterol ratio by substantially increasing HDL cholesterol while lowering LDL cholesterol. They also don't appear to cause diabetes, something that statins can do. But the promise did not pan out. Evacetrapib was safe and efficacious. It was safe in the sense that it did not cause serious side effects such as muscle damage that statins could occasionally do. It was efficacious at making dramatic improvements in the lipid profile. But in phase 3 clinical trials, there was no reduction in major cardiac events. I think this is a pretty good argument for the theory that we have been shooting the messenger all these years. All these clinical trials for statin drugs have a serious weakness. People who have adverse reactions to the drugs are dropped from the study. If these adverse reactions were random, it would not compromise conclusions so much. But what if the people who have the adverse reactions are also the ones most likely to develop heart disease? Then you would end up comparing your control group with a treatment group containing fewer high-risk individuals. These studies were performed to see if statin intolerance could be overcome in order for patients to resume statin therapy. Although I'm not fond of the goal, I do think the studies had some interesting results. In the upper study, coenzyme Q10 ameliorated statin-associated muscle symptoms. My interpretation would be that these patients had marginal levels of coenzyme Q10. This molecule, found in mitochondria, aids energy production and is part of the endogenous antioxidant system. People with lower levels of coenzyme Q10 would be at greater risk of heart disease. The second study evaluated vitamin D therapy to reverse statin intolerance again. Vitamin D was efficacious in doing that. People with lower than normal levels of vitamin D are at a greater cardiovascular risk. So I suspect that statin intolerance is not random, and elimination of patients with low vitamin D or coenzyme Q status could compromise analysis of results of statin drug trials. But there is another finding that I find quite interesting. Look at the dosages of vitamin D3 that were given to these patients. For 12 months, 50 to 100,000 units per week. If these dosages are tested to reverse a drug intolerance, why can't they be tested for their efficacy in treating potentially life-threatening infections with COVID-19? The vitamin D levels did not reach toxic levels, and there were no significant elevations in serum calcium levels or evidence of damage to the kidneys. This slide repeats much of what I just said, but in addition, you should be aware that low levels of estrogen in women and testosterone in men are associated with increased risk of Alzheimer's. Low cholesterol is associated with greater risk of hemorrhagic stroke. In the elderly, it is associated with impaired cognition. A drug approach can have many unexpected side effects over the long haul, especially if the target of therapy is the messenger and not the culprit. This misguided approach has not only distracted from what I feel is the real culprit, insulin resistance, but has potentially placed our population at greater risk by further compromising an already low vitamin D status. Here is another group of drugs that I believe are problematic, especially during this COVID-19 pandemic. Patients with hypertension or diabetes are often treated with angiotensin-converting enzyme inhibitors and angiotensin II type 1 receptor blockers. ACE inhibitors and ARBs can increase level of expression of ACE2, which is found on epithelial cells of the lungs, intestines, kidneys, and blood vessels. As it turns out, ACE2 is a co-receptor for COVID-19. Increased expression of ACE2 could theoretically facilitate infection with COVID-19, which could explain the higher risk of serious COVID-19 infections among patients with hypertension. If the hypertension is being adequately controlled by the drug, what is causing the increased risk? There are better ways to lower blood pressure, certainly much better than eliminating it via a fatal infection. There is a lot of hope that a vaccine will be the answer, but vaccines are not without their risks, and there are certainly many people dying each year of influenza despite the development of a vaccination program. There are risks of hypersensitivity reactions, but those can be minimized somewhat using a vaccine that is free of allergenic substances that do not contribute to the desired protective response. Viruses evolve. Evolution is real, and the genetic drift, the result of accumulating mutations in the viral genome, can result in a new variant that is no longer controlled by the vaccine. A third problem that can sometimes occur is antibody enhancement. I'll discuss that on the next slide. Antiviral drug therapy has its challenges. Rapid microbial evolution usually results in the development of resistant variants, especially when there is selective pressure during the treatment, which there always will be. The virions that remain after treatment are more likely to have developed some form of resistance. Vitamin D, on the other hand, is still efficacious after millions of years of use. Its effectiveness is not based on a single reaction, but on a host of activities. In veterinary medicine, we have some serious coronavirus pathogens. In the cat, there is a feline enteric coronavirus that can cause gastroenteritis, but it is of low virulence. However, this virus can mutate in the cat and become lethal. What happens is that the virus develops the ability to replicate in monocytes. These cells normally help eliminate the virus by means of phagocytosis. But once productive infection can occur in these cells, the infected cells spread the virus throughout the body. The disease that results is called feline infectious peritonitis, and there is no survival. Here is a cat suffering from it. The abdomen is greatly distended with fluid. Certain vaccine candidates have greatly increased the risk of this evolution occurring. This is through the development of enhancing antibodies that fail to neutralize the virus, but actually promote infection. This phenomenon is also seen in a virus of humans called dengue. We already have human coronaviruses that are among the viruses that can cause the common cold. If a vaccine caused the production of antibodies that would enhance infection of either COVID-19 or other coronaviruses rather than neutralize the virus, there is the potential of more serious disease. Some people may have antibodies against the lower virulence human coronaviruses now. It is possible that we may be seeing the result of enhancing antibodies, although I doubt that that's the case. The data I have described indicates an association between vitamin D status and the risk of severe COVID-19 infections. Did I cherry-pick studies showing a benefit of vitamin D supplementation and efficacy in the prevention of influenza and other respiratory infections? To some degree I did. There are some trials that failed to show a significant benefit. In some, dosage may have been too low to substantially increase vitamin D levels. This would be particularly likely if vitamin D status was fairly good to begin with. But importantly, no trials that I know of revealed a danger associated with vitamin D supplementation. We can't be sure that vitamin D will work as hoped, but the low toxicity and high potential benefit to overall well-being would, at least in my opinion, make it a no-brainer. I'm going to briefly address a few other potential benefits of higher doses of vitamin D. Here are several papers addressing a potential cancer preventative effect of vitamin D. Preventing cancer sure beats having to treat it. In the first paper, low levels of vitamin D are associated with a substantially increased risk of various common cancers, including those of the colon, prostate, breast, liver, and bladder. The second paper looked at postmenopausal women who increased their vitamin D intake by 1,100 units. They reduced their risk of cancer by 60 to 77%. The third paper is quite remarkable and is in part why I feel comfortable with taking considerably higher levels of vitamin D. In this study, there was an 82% reduction in the incidence of breast cancer for women with a 60 nanogram per mil or greater level of vitamin D as compared to those with levels lower than 20 nanograms per mil. That blood level of 60 nanograms per mil was considerably higher than recommended by the Institute of Medicine. So why does the Institute of Medicine recommend such paltry amounts of vitamin D? They chose a level that would supposedly achieve 25 hydroxy D levels of 20 nanograms per mil in 97.5% of our population. But they made an error in their statistical analysis, which I won't take the time to explain. But they also assumed that 20 nanograms per mil is sufficient. I think I have shown you plenty of data to seriously question that assumption. The 2018 breast cancer study would suggest striving for 60 nanograms per mil or more. But it is possible to get toxic levels of vitamin D. What are the consequences? Blood calcium levels will be elevated. This can potentially result in kidney stones and inappropriate calcification of soft tissues. As I mentioned earlier, vitamin K2 can help reduce that risk. A blood level of 150 nanograms per mil appears to be toxic. Remember the statin intolerance experiment? 50 to 100,000 units per week for a year did not result in what are thought to be toxic blood levels, and no kidney damage or elevated blood calcium levels were found. This paper, published 13 years ago, reported no toxicity in trials conducted with dosages of 10,000 units or more. 10,000 units sounds like a lot of vitamin D, but it is actually only 1/4 of a milligram. A 100,000 unit dose of vitamin D would just be 2.5 milligrams. The potential toxicity of vitamin D would depend on several factors. One would be how long you maintain that dose. For a single dose, 100,000 units would be safe. But if you took that dose daily for many weeks, you could be reaching toxic levels of the vitamin, causing your blood calcium levels to increase or the development of kidney stones. As this chart indicates, there are ways to decrease the risk of toxicity. One is to drink more water, and another would be to consume less calcium. These are the strategies that Dr. Cicero Coimbra in Brazil has been using to treat his multiple sclerosis patients. He also monitors blood calcium and parathyroid hormone levels. The daily doses he uses can raise vitamin D levels well above what is considered a toxic threshold of 150 nanograms per mil. I mentioned on this slide another important cofactor, vitamin K2. Remember the importance of vitamin K2 in activating osteocalcin and matrix GLA protein, helping make sure that calcium goes to the right place. In this report from India, 62 patients received multiple vitamin D injections of 600,000 units each. Not surprisingly, they developed clinical signs of vitamin D toxicity, which included weakness, constipation, abdominal pain, nausea, vomiting, loss of appetite, small amounts of urination, and altered senses. Serum calcium level was elevated, and the mean vitamin D level was around 125 nanograms per mil. That level is lower than the 150 nanograms per mil level that's reported to be toxic. 3 take-home messages. 1. Make sure your vitamin D3 supplement is from a reliable source. 2. Don't take such ridiculous amounts of vitamin D3. And 3. It's probably wise to keep your vitamin D level under 100 nanograms per mil, probably in the 60 to 80 nanogram range. The U.S. drug approach of lifelong treatment of symptoms of chronic diseases, or in the case of statins, a preventive strategy that may actually contribute to the illness rather than prevent it, has been a mainstay to the health of the pharmaceutical industry, not to the health of the general public. I sincerely believe that addressing common nutritional deficiencies and excesses can make dramatic improvement in the general health and well-being of our nation, and at the same time greatly decrease the cost of our health care. We also need to seriously reconsider the impact some of our health recommendations have had. We have a growing problem of insulin resistance, and a high-carb, low-fat diet is not the solution for people who can't control their blood glucose levels using that approach. The wrong kind of fat is a problem, but it is not animal fat, an important source of fat-soluble vitamins. It is these imbalanced, nutrient-depleted oils that were introduced to our diets in just the last 100 years. We are an inflamed nation. Tempers are rising, as well as the death toll from the pandemic. Here are just a few of the conditions in which inflammation plays an important role. I mentioned Alzheimer's, but mental illness can be another outcome. The brain is an organ, and just as inflammation damages our other organs, it damages the brain. I remember reading a report concerning violence in a prison. Changing the diets of the inmates resulted in a significant decrease in violent events. Uncontrolled inflammation is a killer, and that fact has become all the more apparent during this COVID-19 pandemic. On this slide, I mentioned some causes of chronic inflammation. Consumption of excessive levels of omega-6 fats found in grain oils leads to excessive production of potent inflammatory mediators. A diet high in sugar and calorie-dense but nutrient-void carbohydrates will deplete our reserves of antioxidants. Just as different fuels vary in how dirty they burn, the same goes for our source of calories for energy. Reactive oxygen species are generated when we metabolize our energy sources. Fats are actually the cleanest burning fuels. Protein is certainly not clean burning. People can survive on a diet of 80% fat or 80% carbohydrate, but lifespan would be pretty short on a diet of 80% protein. The Inuits in the far north, when they consumed their traditional diets that were very high in animal fat, enjoyed good health. That high-fat diet would have helped them maintain a sufficient vitamin D status at such a high latitude. But it would also have spared their antioxidant stores. They certainly would not have gotten much vitamin C in their diets. For some obese people, a diet consisting mostly of nutrient-dense carbs, such as salads and greens, can bring substantial health benefits. But these diets are low in calories, and the body turns to its own fat stores as a major energy source. And that is a good thing, because fat is a clean-burning fuel. I mentioned taurine. Taurine is an amino acid, but it is not part of proteins. Like vitamin D, taurine has many roles. It is particularly important in maintaining the health of the heart, kidneys, and eyes. We can produce our own taurine, but not enough to maintain optimum levels. This is the same as seen in other species. Cats, throughout their evolution, have relied on a diet for their taurine. And if their diet is deficient in taurine, they develop a condition known as dilated cardiomyopathy. The heart becomes greatly enlarged but thin-walled. As omnivores, we do not need as much taurine in our diet as a cat, but it is still an important nutrient for optimum health. Like vitamin D, taurine plays an important role in controlling the inflammatory response. Good sources of taurine are the dark meat of poultry, liver, heart, fish, and shellfish. Many in this country have limited their consumption of the dark meat of chicken in favor of the white meat. The dark meat has about 5 times as much taurine as white meat. I would strongly advise against the use of energy drinks as your source of taurine. Many doctors are struggling with the best approach to treat cases of COVID-19 infection, and some anecdotal evidence suggested that we tread carefully. In France, one doctor reported about 4 children who took a turn for the worse when they were given ibuprofen. Subsequently, the French health minister and the World Health Organization issued a warning. The British Pharmacological Society suggested acetaminophen as the first drug of choice. Then the World Health Organization retracted its earlier warning against using ibuprofen. One worry about ibuprofen is that, like the ACE inhibitors I mentioned previously, it causes an increase in ACE2, a co-receptor for the COVID-19. The verdict is out. on the best strategy to manage COVID-19 cases. But I am concerned about the suggestion that acetaminophen should be a drug of choice. There's pretty strong evidence that we are experiencing an epidemic of autism, and the major cause is an over-the-counter analgesic called acetaminophen, better known as Tylenol to most of us. I will briefly digress to give that statement support. Almost every mother has given her child children's Tylenol, and I'm sure that during this COVID-19 outbreak, many more children will be exposed to this very dangerous drug. I believe that we as a nation tend to overtreat, but underprevent health issues. We also seem to have very little tolerance for pain or discomfort. We are currently suffering a terrible opioid epidemic, in part because of a rather careless approach to pain management. But the opioid epidemic is not the only epidemic that we are suffering because of overtreatment of pain or discomfort. Fever, if not too high, can play an important role in fighting an infection. When we treat a mild case of fever in a child for the sake of improving comfort, it can have adverse consequences, both short-term and long-term. The lower chart from this paper documents the dramatic increase in the incidence of autism that has occurred since the 1980s. There has been a shift in the timing of the onset of symptoms, and the greatest increase in incidence has been in the development of symptoms at about 1.5 years of age. The genetics of populations do not change rapidly, certainly not in the span of a few years. This data indicates an environmental influence as the cause. To make a short story short, there are a number of things that could be increasing the incidence of autism, including a deficiency of vitamin D. But acetaminophen stands out. Its use to treat mild to moderate discomfort in young children has steadily increased since the early 1980s, when fear of aspirin-induced Reye syndrome in children prompted a change in analgesic preferences. The incidence of autism in boys is about 4 times greater than it is in girls. One likely reason for that is that male infants are more susceptible to oxidative stress than female infants, and are more susceptible to toxin-induced oxidative stress. Further evidence for a connection between oxidative stress and autism is seen in the case of Down syndrome. Down syndrome is associated with a substantial increase in oxidative stress and inflammation, and more than 5% of people with Down syndrome have autism. Getting back to this slide, analyzing the records of over 300,000 boys followed for 9 years, there were nearly 5,000 cases of autism. Boys who had been circumcised were twice as likely to develop infantile autism before 5 years of age. These boys were born between 1994 and 2003, when acetaminophen administration had become the standard analgesia protocol for neonates. This study looked at the correlation between the prevalence of autism per 1,000 males in a country or state and a percentage of the male population that was circumcised. A correlation coefficient of 0.95 is extremely high, but circumcision is nothing new, and it alone certainly would not explain an epidemic of autism. So what would be the mechanism? Acetaminophen is metabolized 2 different ways. Young children before the age of 2 are limited in their ability to metabolize the drug, primarily because they have an underdeveloped glutathione-dependent system. The end result is that acetaminophen depletes the low levels of glutathione they have, and the children suffer oxidative damage, including damage to the brain. In addition, acetaminophen inhibits production of a couple prostaglandins, one that may play a part in brain development, and another that may play a role in controlling inflammation in the brain. Whatever the mechanism, studies have accumulated that implicate acetaminophen deeper and deeper in this mess. So the theory is that a combination of increased inflammation and oxidative stress and reduced ability to handle that stress leads to autism. That reduced ability to handle oxidative stress is definitely both genetic and environmental. Polymorphisms in the glutathione pathway impact risk. A vitamin D deficiency combined with early exposure to acetaminophen in the neonatal period and a genetic predisposition would be a perfect storm inducing autism. Giving a child acetaminophen during his first week of life outside the womb, at a time when he is ill-prepared to handle the drug, would likely be the trigger responsible for the autism epidemic, especially if that child is genetically less capable of controlling oxidative stress, as is the case with young male children. It could also be a perfect storm for susceptibility to the cytokine storm of a COVID-19 infection. There have been a number of reports of signs of autism appearing shortly after vaccination. In this paper, using a parent survey and comparing autistic children with normal children aged 5 years or less, it was found that acetaminophen use after vaccination for measles, mumps, rubella was significantly associated with autistic disorder. The autistic children were 6 times as likely to have received acetaminophen as the control children. No association was found for ibuprofen use after MMR vaccination. Can acetaminophen be used in a safer manner? Well, yes, it can. We know that depletion of glutathione is what creates such a vulnerability, especially to oxidative damage by the highly toxic metabolite of acetaminophen. One way to help prevent the depletion of glutathione and subsequent oxidative damage would be to provide a precursor, N-acetylcysteine, to restore glutathione levels. Overdose of acetaminophen is the most frequent cause of acute liver failure, and N-acetylcysteine can prevent the extensive liver damage. When a child has autism, there is an approximately 20% chance that the next sibling will also develop autism. In this study, 19 mothers of children with autism were given 5,000 units vitamin D daily during pregnancy. Newborn infants were prescribed 1,000 units vitamin D daily until their 3rd birthday. Only 1 out of 19 developed autism. There is an emerging syndrome occurring in children that is thought to have been precipitated by an infection with COVID-19. It is characterized by widespread inflammation. I don't have all the details, but at least some children are antibody positive for the virus, but the virus can no longer be detected. This suggests a delayed response to the virus. There's typically persistent fever. I don't know how that has been treated, but it would not surprise me that repeated doses of acetaminophen were given. A panel of experts speculated that the acquired immune response activated an inflammatory response in genetically susceptible children. That sounds a little familiar. If a child has spent the last 2 months indoors, I doubt that his or her vitamin D status has improved since the pandemic began. Hopefully, vitamin D plus N-acetylcysteine will be considered in the therapeutic regime for these children. Here is another nutrient that has been seriously neglected during this pandemic. I take a daily dose of vitamin C. If I were to start showing any signs of a respiratory infection, I would certainly take much larger doses for at least a few days to a week, and the form I would take would be liposomal, which can be absorbed much more effectively and achieve considerably higher blood levels. Most mammals and other animals produce their own vitamin C, and when confronted with a stress such as a viral infection, they produce much higher levels of it. We and other primates have the gene necessary to produce it, but we have lost the ability to express it. Guinea pigs and fruit bats also cannot produce it, but they get plenty of it in their diet. Like us, they can get a sufficient maintenance dose through their diet. But unlike other mammals, they cannot greatly increase their vitamin C levels in response to an infection. Perhaps that is why guinea pigs have been a good model for many human infectious diseases. Most of us think of vitamin C as an antioxidant, but it is also essential for the production of collagen. A severe deficiency causes scurvy. Blood vessel walls deteriorate and start leaking blood. It doesn't take much vitamin C to prevent scurvy, but how much does it take to prevent aortic aneurysms or fight infections? There is a very interesting book by Thomas Levy that provides a long history of the use of vitamin C in earlier days. There have been some pretty amazing anecdotal reports of cures of people on the brink of death from various infectious agents and toxins. What is amazing to me is that very little follow-up of research has been done to evaluate these putative cures, and those studies that have been performed have used much lower doses than originally reported as successful therapies. There has been much skepticism from medical practitioners, but little action to seriously evaluate efficacy. I mention an anecdotal case on this slide and provide a link to a YouTube video that describes the amazing recovery of a man on the brink of death with swine influenza. I think it is of value to check it out, not only for the results, but also to see just how difficult it was for the family to get the doctors to try this very inexpensive and relatively safe treatment for a comatose man who is about to be taken off life support. I am sure that I will be ostracized by some for bringing this up, but I have pretty thick skin. Well-designed studies with truly high doses need to be performed. In China, a trial of vitamin C infusion for treatment of severe COVID-19 pneumonia has begun. The dosages are still much lower than have been used in successful treatment in the past, but I am encouraged that this approach is being given serious consideration someplace, if not here. A vitamin D trial is also planned in Spain, but I doubt that a single dose of 20,000 units will make a big difference in vitamin D status, especially if done in the summer when vitamin D levels are higher. If you've made it this far, you have been very patient. I can get carried away with some topics, and as far as I know, you have been a captive audience, but I will try to wrap this up. We have been hearing how warm temperatures, low humidity, and direct exposure to the UV spectrum of sunlight will reduce infectivity of COVID-19 and control the spread of the virus. But this virus spreads easily from person to person, and most close encounters occur indoors. If vitamin D status is actually a critical ingredient in the recipe to reduce susceptibility to this virus, we need to make sure we have what the recipe requires. We will not have improved our vitamin D status by staying indoors these past 2 months. I think, as tragic as this pandemic has been, it may bring about much-needed changes in our approach to health care that could save millions of lives and restore good health for many in the near and distant future. In my teaching days, I would often use this slide, much to the chagrin of my students. He is the bridge keeper from the movie Monty Python and the Holy Grail. In order to cross the bridge, you would have to answer 3 questions. We have many more questions to answer than that, but before we answer them, we need to start asking them. For example, we know that in order for vitamin D to be active, it requires the addition of 2 hydroxyl groups. The first is added in the liver. Liver function could be compromised in a comatose patient. Perhaps giving calcitriol, the active form of vitamin D, would be more effective in those patients. We come in all shapes and sizes, and one size fits all is not going to be the best approach. For as long as we have known about vitamin D, it is unfortunate that we still have not fine-tuned optimum dosages for people of all sizes, shapes, and conditions. Infections can often become a mixed bag. For example, viral infections can be complicated by secondary bacterial infections. This paper deals with an experimental model to study the SARS coronavirus. If guinea pigs were infected with the SARS coronavirus alone, the lungs would become inflamed, but it would not be fatal. If guinea pigs were infected with a real virus, death would occur within 22 to 30 days of post-inoculation. However, if the guinea pigs were given both the Rio virus and the SARS coronavirus, there would be 70% mortality within 4 to 7 days post-inoculation. The point is that next winter we could be facing two different respiratory pathogens or more circulating at the same time. And we may face some cases of dual infections with unpredictable outcomes. When we come up with a vaccine for COVID-19, will it be that effective in those at greatest risk of infection? If not, can we make it more effective by improving vitamin D status? This paper, using a mouse model, demonstrated that supplementation at time of immunization against influenza virus improved the mucosal antibody response in mice deficient in vitamins D and A. Vitamin D does its work regulating gene expression as a complex with the vitamin D receptor. We know that there are variations in that receptor and that people with certain VDR variants are at greater risk of developing certain infectious diseases, such as tuberculosis and leprosy. Will people with certain VDR variants be at greater risk of developing severe COVID-19 infections? If so, can that risk be reduced by simply increasing the preventive and/or treatment doses of vitamin D? I hope one lesson that we learn from this tragic pandemic is that we start to listen to scientists instead of the opinions of totally ignorant individuals who know nothing about issues threatening our health or the health of our planet. There has been active suppression of the voices of scientists, especially concerning the threat of global warming, and that has got to change. Hopefully people will not only listen to the evidence, but also heed the advice based on that evidence. By now, we should appreciate the dire consequences of delaying our response to a serious threat. I can't emphasize enough how important it is to have independent research. The incentives for some companies to muddy the waters can be great. I do think our doctors are missing out on much of the latest research that is not drug-oriented. Many of them are enthusiastic and knowledgeable concerning alternatives to conventional drug therapies. But if standards of care don't reflect more natural preventive approaches, most will hesitate to stray far from what is currently expected. Consider your sources. PubMed has a treasure trove of research articles at your disposal. The more active a role you take in your family's health, the greater chances you will all have to weather this COVID storm and the storms to come. Choosing the wrong leader can take us down a very dangerous path. Thank you.